Hello everyone, we are starting off the lecture with anatomy of cornea. See, this outer layer over here that you can see is cornea. Now this layer cornea is made up of many small layers. We'll study in detail about each and every layer that is making up the cornea. So starting with this diagram, see over here the topmost layer is the epithelium layer. Second one below epithelium will be the basement membrane. Third one is Bowman's layer. Now we'll learn something about each layer. See, this epithelium layer can regenerate. Why? Because the bottom layer in the epithelial layer that you can see that divides by mitosis and it will replace the dead cells. So this epithelium layer can regenerate. While the Bowman's layer below it, it is acellular. Means there are no cell. If there are no cell, then who will do the mitosis and regeneration, right? The Bowman's layer is acellular. So whenever there is any trauma over here, then if the trauma is subsided only to the epithelial layer, then there will be regeneration and there will not be scarring. But if the Bowman's membrane is broken down by the trauma, there will be scarring. The third layer we will see is the stroma. See, epithelium and the basement membrane and Bowman's layer is done. Third one is stroma. In the stroma, you can see that it is occupying somewhat 90% area of the cornea and it is the strongest layer. Now here we have descent membranes and above it here over here we will have the Duas layer. So we'll have Duas layer and the descent membranes layer. Duas layer is the strongest corneal layer, okay, not the stromal layer. Duas layer is the strongest layer. Then below it we have descent membrane which is secreted by the endothelial cells. So these endothelial cells will secrete this descent membrane. And the last we have single layered endothelial cell. This is the most important layer of the cornea. Now we'll study in detail about each and every layers. Before that we'll just quickly revise the layers. First is epithelium, then the basement membrane below epithelium, then we'll have Bowman's layer, then stroma, then we have two Ds, Duas layer and Descemet's membrane, and at last we have endothelium layers. These all features we'll study in detail about each and every layers. Now epithelium. See, let's first look at the type is non-keratinized. Why? Because the light has to go through the cornea, right? So there cannot be keratin is in hair, skins, etc. So the first thing, it is non-keratinized stratified squamous. So why does this layer has to be multi-layered? See, this layer is multi-layered layer. Why a single layer can't work? No, because the cornea is the outermost layer of the eye. It does the protection job, so we cannot just put a single layer. So that's why we have stratified squamous or multiple layered. So the superficial layer has microvilli. See, here you can see these are microvilli and the squamous cell with microvilli. Why? Because in the superficial layer, let's say this is the superficial layer, there will be tear attachment. Tear will get attached. If the tear doesn't get attached, tear goes down and there will be dryness of cornea. So to hold or attach this tear, they have the microvilli. So the microvilli will get attached to the tear, attached to the mucin layer of the tear film. See, this is an entire tear film and the this layer is mucin layer which is getting attached to the cornea. So that's why we have this microvilli. Now the basal layer, I told you that this epithelium can regenerate. Why it can regenerate? Because the basal layer are columnar cell which do mitosis, and due to this mitosis they replace the cell which are damaged. So the basal layer is responsible for regeneration. So we have superficial layer with squamous cell and microvilli, and the basal layer is columnar cells which does the regeneration. Next layer is Bowman's layer. I told you Bowman's layer is acellular. If there are no cell, there is no one to regenerate. So it is acellular and does not regenerate. And it heals by scar. I told you if there is a trauma limited to the epithelial layer, there will be healing and there will not be any scar because the basal layer will do mitosis and regenerate those lost cells. But if the Bowman's membrane is injured, then we'll have a corneal scar, opacity and loss of vision. So let's learn in detail about corneal opacity. See, this over here you can see this is a corneal opacity. Now first of all, let's learn the basic. What is a pupillary margin and iris? See, first of all, this is the cornea. As I told you, this is not iris. Above iris and pupil, we have cornea, but we can see pupil and iris. Why? Because cornea is completely transparent. So through it, I can see pupil and this iris. In this diagram, you can see this is the cornea, and behind it, this is iris and pupil. So I can see this iris and pupil completely because this is transparent. But if there is any injury, then there will be scarring, and I will not be able to see this iris and pupil if this is not transparent, right? So in here we have two things: pupillary margin and iris details. What is pupillary margin? This margin blackish over, this margin over here, you can see this is the pupillary margin, and these small details of the iris, the crypts, etc., these are iris details. Now if there will be any opacity, then I will not be able to see what? Pupillary margin and iris details. So this is the grade of opacity. See, over here there is only little bit of opacification, here there is somewhat more, and here there is complete white spot. So we have 1, 2 and 3. What are these names? This is nebular, macular and leucoma. What? Nebular, macular and leucoma. Now why this nebula is faint? Because nebula, the damage is only till here, superficial layer of stoma and Bowman. So Bowman's and superficial stoma, only this much is the damage. But in macula, it occupies less than half of the stoma. So the damage is down here. And in leucoma, it is greater half of the stoma, more than half of the stoma. So that's why these three are different. Now, which is the faintest? Nebula is the faintest and most opaque is leucoma. Let's learn something in deep about this. See, here in nebula it is faint, so we can see both pupillary margin and iris, both we can see through it. Here I can see this pupillary margin and also this small iris details. But in macula I can clearly see this blackish pupillary margin, but I cannot see any iris details. And in leucoma you can clearly see I cannot even see this pupillary margin. So this is how the severity is graded. Now there is an question. Now, which will have the maximum vision loss and maximum visual discomfort? There are two things: visual loss and visual discomfort. Visual loss is you cannot see anything. Visual discomfort means there will be some glare or something, right? So in nebula, I can see that there is only a bit of opacity. So the light can pass through it. Light can pass through this opacity, but light will scatter. So in this image I can see this is the normal vision, and this is the vision due to scattering of light. So in nebula the opacity is so faint that it will allow some light to pass through it, but that light will scatter. So I will have maximum visual discomfort in nebula. But in leukemia the opacity is so dense that light is not passing through it. If the light does not pass, then there will be no glare or anything, right? So leucoma will have maximum visual loss, right? And nebula has astigmatism. What is astigmatism? Normally when the light comes, it falls at a single focal point on the retina. But in astigmatism, there are multiple focal points, so there is a hazy image, right? So in nebula, due to this astigmatism, we have maximum visual discomfort. We'll see all this type of glares and all, right? So now coming back to our layers, we discussed epithelium, then basement membrane and Bowman's layer. In the Bowman's layer, if there is a injury over here, then we have different type of corneal opacities. Now we'll study about stroma. See, stroma is first made up of what? Let's say these are the collagens. I'm making a random, I'm not making a random arrangement. Collagens are arranged regularly in a pattern. This will allow the light to go through it. So if this regular pattern of collagen is disturbed, then there will be no passage of light through this cornea, and it will make cornea opaque. Normally cornea is transparent. So we have first collagen, which is most common, is type 1 collagen arranged regularly. Second is glycosaminoglycans. See, what are glycosaminoglycans? In the stroma we have some sponge like glycosaminoglycans. What does they do? Whenever there is a bit of water or excess water here, these glycosaminoglycans, they absorb this water or they take this water. So we don't want any water in the stoma. Why? Because if there will be any water, then the stoma will not be transparent, it will be opaque or translucent. So these glycosaminoglycans are like sponge, they will take away the excess water. So the most common one is keratin sulfate, and it is responsible for what? Corneal transparency. Why? Because they will take the water away. So stroma is made up of collagen and glycosaminoglycans. Now the next layer is Descemet membranes. We have already studied that the Duval's layer is just the strongest layer. There is nothing in detail about it. The Descemet membrane over here, we know that it is the endothelium which divide and make this layer. So it is secreted by underlying endothelium layer. Now in this image, let's see this is the pupillary margin, these are iris details, and over here this covering is the cornea which is transparent, so I can see both this structure. Now at limbus is corneoscleral junction. Over here the cornea is over, and from now here the sclera will start. So the limbus is the corneoscleral junction. So now this Descemet membrane, let's say this layer is taken from here, right? That epithelium will be here and Descemet membrane will be here, right? So put this diagram over here, then this Descemet membrane will be present only till here. This is Swebeck's line. Beyond this Swebeck's line there is no Descemet membrane, right? So the peripheral termination of Descemet membrane is this Swebeck's line. So this was all about Descemet membrane. Now the last layer and the most important layer is the endothelium. Why most important? Because let's say there is some water over here in the stoma. How will we clear this water? First was that these glycosaminoglycans were taking this water. Second is this endothelial cell. Let's see this endothelial cell. They have a sodium potassium pump. So they'll throw the sodium out like this. So the sodium will go, let's say, in here inside the aqueous chamber, and the water will also follow this sodium. So this endothelium is having sodium potassium pump which is responsible for maintaining the corneal transparency. Most important layer which is responsible for maintaining corneal transparency is the endothelium. But the problem is that endothelium is a single layer cell, like, okay, and it cannot regenerate. So after the age passes there will be wear and tear and the cells will degenerate, and the amount of cell will decrease over time because it cannot regenerate. So initially the cells are up to 3000 to 2400 cells per mm cube. This is normal, but when the cell decrease, and we know that it is a single layer, right? So when the cell is decreased, now these two cell, there is no cell in between them because it has degenerated. Now either this cell has to increase its size, which is polymegathism, or increase or change its shape, which is polymorphism. So this cell has to either increase its size or change its shape so that it can fill this gap and there is no water leaking over here, right? So let's say this, there is this cell injury. Now if the surrounding cell don't change the size or the shape, there will be water coming inside the stroma. So at this cell decrease count, whenever the cell decreases by 2400, there will be polymegathism and the polymorphism, right? And critical point is less than 500. If their cells are less than 500 per mm square, then there will be edema, water will come over here in the stroma. So we have corneal decompensation, edema and hazy cornea, and there will be loss of vision, and these cells are counted by specular microscopy, right? So if we want to transplant this cornea, then the most important thing that we look over here is the cell count. If there are less cell, then we don't transplant this cornea to any other person. Now the nerve supply, see, now I want to see the sensory nerve supply of the cornea. So let's look at this image. This is the trigeminal ganglion. From this trigeminal ganglion or trigeminal nerve there will be ophthalmic nerve, and from ophthalmic there is nasociliary nerve. So we have trigeminal, then ophthalmic, and then nasociliary nerve. Now this nasociliary nerve will be having sensory supply to this eye. Now what is sensory supply? Let's say I take a cotton ball and I touch over here in the cornea. So this sensory sensation will go through nasociliary, ophthalmic and trigeminal ganglion towards the brain, right? So this is what corneal sensation test, that the sensation is passing from cotton wisp to cornea to fifth afferent nerve, which was trigeminal nerve, to the brain. From brain, the afferent is the seventh cranial nerve, and this seventh cranial nerve will send the signal to this muscle, which is orbicularis oculi, and this muscle will shut the eyes closed, so there will be blinking. So whenever there is cotton spot over in the eye, we know that the eye blinks. Why? Because the pathway is, let's revise the pathway again. The sensation goes here, from there it goes to the nasociliary, ophthalmic, trigeminal to the brain, and from brain it comes by a seventh now orbicularis oculi, and there will be blinking. Now in which case there won't be any blinking? In the case of corneal anesthesia, when there is a pathology in herpes, diabetes and leprosy. Herpes, diabetes, leprosy, they have corneal anesthesia, and this eye won't be blinking via the corneal sensation. Now, some general properties about cornea. See, cornea is a convex. We know that cornea bends light, right? So it's a convex converging. Curvature is equals to power. More the curvature of cornea, more the curvature of cornea, more will be the power. Now, color. Cornea is transparent. As we know, it is transparent, so that's why we can see this iris and pupil through the cornea, right? So this was all about some general properties of cornea.